Okay, so we have Liz Cray, Pink, and Kaylee Composto.
Touch anything.
Okay, go ahead.
Okay, so Kaylee and I are proposing the Chelsea and Childy project. Chelsea and Childy are factors involved in the Salmonella virulence cascade. Brief background for anyone who forgets their Bio 1/ never learned about transcription factors. Transcription factors are proteins that bind to specific sites in the DNA and initiate transcription of, usually upstream of the gene of interest, you can see here, and initiate transcription, and as you know, eventually transcription follows. So you go from DNA to protein, and without transcription factors, this could not occur. So honestly, they're vital for proper cellular function. So this project originated, kind of stemmed off of another project in Dartmouth called the TOPS-T project. And we actually have a couple of old smart team people here, yeah. So they'll know about it, but can you slide? So TOPS-T is a member of the Araceae family, superfamily of proteins, as are Chelsea and Childy. Researchers at Dartmouth expressed purified salt crystal structure of TOPS-T, and when they did so, they noticed something unusual. They realized that in the presence of certain fatty acids, when they were bound to the greenest TOPS-T, when the fatty acid is bound to TOPS-T, TOPS-T cannot bind to the DNA, and thus cannot initiate the transcription of the cholera virulence cascade. So with the fatty acid, no cholera. So honestly, they're not good answer. This could be used as for novel drug, remedy, novel drug treatments, etc. Since Chelsea and Childy are also Araceae superfamily proteins, the current hypothesis is that maybe Chelsea and Childy work in much the same way. So, Chelsea and Childy are self-regulating transcription factors that also regulate HIL-A, the primary regulating toxicity factor in Salmonella. So, even though HIL-A is most directly analogous to TOX-T, without Chelsea and Childy, HIL-A won't be transcribed, thus still got no Salmonella. So, the overall goal of the project is to express, purify, and crystallize Chelsea and Childy. What the crystals is to solve the structure of Chelsea and Childy. Once we have the structure, we'll be able to take a better look at the biological mechanisms of infection and see whether or not they mimic that of TOX-T and cholera. If they do, this could lead the way for future drug studies and other new biotreatments for Salmonella.
Can I ask a question? I don't know how we're doing this, but I have a question.
Yeah, okay.
HIL-A, is it an Araceae family regulator? Is there a reason why we wouldn't look at that?
Good question.
It may not be, and I'm just wondering. Because if it's not Araceae, then we don't really care. But.
We tried to look that up. I think that, I can't say for sure, to be honest. For whatever reason, we started doing Chelsea and Childy, and I would assume that because HIL-A is an Araceae, but I can check that out.
I would also assume that, but it would probably be.
It would make the most sense. There would be no other reason not to look at HIL-A if it weren't Araceae. So I'm assuming Chelsea and Childy.
Because if you're saying that HIL-A is analogous to TOX-T in terms of being a primary toxicity regulator,
right, then
why not HIL-A? But
right. Yeah, no, exactly. But right. I assume it's because it's not Araceae. Mm-hmm. If it's not Araceae, then
just look into it.
Yeah,
What have you solved so far? Like, if you go back to the previous slide.
Yeah.
What here is known? What is?
Chelsea and Childy are not known. Like generally, like their function is known. I'm not sure it's not known. Mm-hmm. I don't believe HIL-A is known.
We
Okay.
don't have structures for any of them.
Okay, and Hill A is your target, or is your
No, Hill C and Hill D are the target.
okay?
Because they're air C.
Yeah. Any other questions for?
So we plan on carrying this out by PCR in both Hill C and Hill D out of vectors PKN1 and PKN2. Then we're going to
Has that been done?
previously, yes.
Okay.
But
All right.
but we're starting fresh this year due to
But you have primer sets. You have everything you need. You don't.
Yeah,
You're not doing this from
that's
scratch.
okay, too.
Okay.
yeah. Then we're going to digest Hill C and Hill D and PTX1 with restriction enzymes and use NDE1 and SMP1. Then we are going to ligate both Hill C and Hill D separately into PTXV1 because they both go into the same vector. Then we're going to transform into BL21 E. coli, so we're transferring it back into bacteria. And then we have to start the more unknown part, which is the expression trials, and find an optimal temperature and time for the cells to grow up in order for them to express.
How are you doing that?
We're going to set, we found through the toxicity that they grow better at lower temperatures. So we're going to play around the span of roughly 16 to 22 degrees in that window.
In what? I mean, what are you gonna do this in? How are you gonna?
We're working on it.
Yeah.
What's your temperature range?
I think we're planning 16 to 22 degrees.
Anything higher than that is shown to not be effective for expressing this particular kind of protein. It doesn't work with analogous ones.
We have a lot of trouble with it being insoluble.
Yeah.
So right now, the way I'm looking at this, the only thing I see that you don't have is a way to regulate some chamber to regulate temperature, right? Like, because
Yeah,
everything
we're-
else, that's gravity, right?
Yeah.
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 Everything
So-
else is fine.
Okay.
I'm not sure that we've really tested room temperature that much, so it's possible that room temperature could be an option. And I think that'd probably be the first place to start, given that we can express at room temperature.
Mm-hmm.
And then, I am looking into incubators on my list.
What kind of price range are we talking?
But I mean, I found some used, it's a couple thousand that I can get, I used before.
Okay.
So,
Next.
so after the expression trials, we're going to try to express Hill C and Hill D. But to do that, we have to purify them and use the gravity flow chitin columns, where Hill C and Hill D will attach to the chitin beads, and then we will use pH buffer in order to purify them. Then we're going to centrifuge them in protein concentrators in order to try to get as much protein in as little amount of liquid as possible before it crashes out. And then we're going to screen for crystal conditions, where we will set up crystal plates in various conditions in order to see which is the optimal condition for these protein crystals to grow in.
Okay. So what kind of like when you're doing these? So you're going to be able to get to most likely the transformations without any real troubles, right?
Oh, yeah.
Here, me.
Okay, and then I guess when the expression trials is kind of where you start to branch out and need to do a lot of replication experiments
Yeah.
and stuff, right? So how many people are we talking? What
We
kind
have
of?
that later.
Okay.
You wanna wait, like, we have all that.
Yeah,
No, she put in her password and
My computer's password.
yeah. All right. Can we move it? I would like to try to keep these under 10 minutes if possible.
Go ahead, go ahead.
So then we move on to crystal analysis, where, as I said earlier, we would set up crystal trays in order to find the optimal condition. And we will, if we get crystal growth, this is ideally what we would be looking for, something of this shape.
But your
that's
main-
not Chelsea and Cody, that's.
This is all off-site, and your main project is up until this, right?
Yes.
Okay, so this is, you have the equipment to do somewhere.
This, yeah,
Yeah.
there's,
Okay.
We can do some screening analysis by sending it away, the protein away. It's not very expensive.
Okay.
and this is far down the road, though, we get as far as purification this year, that'd be great.
Okay, so then let's move
Yeah,
forward.
this is just for further education. Yeah.
So for our budget, in order to purify protein, we have to use bottle top filters and then put that through the chitin column in order to purify. So 0.2 micrometers is the filter that is typically.
Did you shop those?
I think we have some,
Okay.
and we can use 0.45 micrometers.
So,
I think we have those.
I mean, okay, so if we're talking, what I want to know is how much is this going to cost in the next 6 months?
Given next 6 months, nothing.
Nothing.
I don't think-
I mean, it's gonna cost our general maintenance for like
Yeah.
DNA stuff,
So stuff that we would
overlays.
just get on top of class anyway.
Yeah,
Exactly.
I don't think, we certainly won't get
This
this
is like the
far.
project as well.
Yeah.
So theoretically, this costs nothing above maintenance.
Until we need a.
Until you do, yeah. Wait, what?
If we get an incubator.
We
Oh,
can
okay.
do
That-
expression trials.
Which I need for class anyway, so really
Right, right, right.
that's also a shared cost.
Okay.
But otherwise, it's pretty general stuff.
Okay.
Yeah, we would need concentrators, I guess that's true, if you can purify the proteins.
And then the next one? Next slide.
Oh, next slide.
People.
So this kind of the skill set we're looking for is we're looking for maximum of probably 2 people.
Mm-hmm.
Otherwise, it's going to be hard for them to learn it. And we figured that Liz will take like Kelsey and I'll take Cody, and we'll each take one person, and they will just do that one transcription factor.
As far as hands go, it's the same skill set though, right?
Yes.
It
Absolutely, yes.
is pipetting and it is transformation and
It's
That's
like
Yeah.
it's, yeah.
how long would it take you to train someone if you had them 2 out of 3 hours a week?
Can we assume they already know how to pipette or?
No, from scratch.
2 or 3 hours a week?
Yeah.
3 weeks? 3 weeks?
3 weeks.
3 weeks?
Yeah.
Not more than
Well,
that.
I don't know about for everything, but you could give them general training.
But,
Like we
yeah,
could
but like
do
up to the point that we need in the next
PCR,
6 months, I mean.
LD.
Yeah.
Yeah.
Okay. Alright, so it would take 9 hours of training
Yes.
and no money until you're doing except for the incubator.
Yes, until purification, no money except for the
Okay.
I don't think we need much for purification.
I mean, purification really won't cost much out.
Okay. Alright. Is that it?
That's
And
it.
yeah.
Alright, thank you.
And
I don't think we need citations.
No.